Welcome everyone to the presentation on Bluescript vectors PBS and PBC. Today, we will explore these vector systems and understand their significance in molecular biology and genetic engineering. Overview of Bluescript vectors. Bluescript vectors, abbreviated as PBS, are versatile cloning vectors commonly used in molecular biology research. They are derived from the plasmid PBR322 and contain several useful features. The PBS vectors are high copy number plasmids, which means they can replicate in large numbers within bacterial cells. Features of PBS vectors. Multiple cloning site, MCS. PBS vectors have a unique MCS that allows the insertion of DNA fragments at specific restriction enzyme recognition sites. Ampicillin resistance gene, AMPR. PBS vectors carry the AMPR gene, which confers resistance to the antibiotic ampicillin. This feature enables selection of bacteria that have successfully taken up the vector. LACZ alpha fragment. PBS vectors also contain a fragment of the LACZ gene, known as LACZ alpha. This fragment can be used for blue-white screening to distinguish between recombinant and non-recombinant clones. Advantages of PBS vectors. Versatility. PBS vectors can accommodate various insert sizes, making them suitable for a wide range of cloning experiments. Easy screening. The blue-white screening method allows researchers to quickly identify positive clones, simplifying the screening process. High copy number. The high copy nature of PBS vectors ensures an abundant supply of cloned DNA, which is advantageous for downstream applications. Applications of PBS vectors. Gene cloning. PBS vectors are commonly used for cloning and amplifying DNA fragments of interest for further analysis. Protein expression. The MCS in PBS vectors facilitates the insertion of genes for protein expression, enabling the production of recombinant proteins. Mutagenesis studies. PBS vectors can be employed for site-directed mutagenesis, allowing researchers to introduce specific mutations into genes. PBC vectors. PBC vectors are a subfamily of PBS vectors with additional features. They possess the same core features as PBS vectors but also include the LACZ delta M15 mutation. This mutation eliminates the expression of the LACZ alpha fragment, which can be useful in certain applications. Advantages of PBC vectors. Elimination of LACZ alpha expression. The absence of LACZ alpha expression in PBC vectors eliminates the need for blue-white screening. This feature is advantageous when working with genes that interfere with the blue-white screening process. Selectable markers. PBC vectors retain the AMPR gene, allowing for selection of positive clones using ampicillin resistance. Applications of PBC vectors. Protein-protein interactions. The absence of LACZ alpha expression in PBC vectors can be beneficial for studying protein-protein interactions using the two-hybrid system. Library construction. PBC vectors are suitable for constructing genomic or cDNA libraries due to their ability to accommodate large insert sizes. Conclusion. Bluescript vectors. PBS and PBC are widely used tools in molecular biology and genetic engineering. Their versatile nature, ease of use, and numerous applications make them valuable assets in research laboratories. By harnessing the power of these vectors, scientists can advance our understanding of genetics and develop innovative solutions to various biological challenges. Thank you for your attention. I hope this presentation has provided you with a good understanding of PBS and PBC vectors. I'm happy to answer any questions you may have.